Okay, in this video we're going to talk to you about how long cannabis is detectable in urine sample testing. We're also going to demonstrate a urine test using a Core Test's cannabis test cassette device. It's a very common question that we are asked: how long after use will cannabis be detectable in urine testing? The answer is there is no straightforward direct answer. Because cannabis is fat soluble and is spread widely throughout the body, it can be stored effectively and come out of the system very gradually after sustained and heavy use. This is why you will see various window periods quoted on different test devices and different manufacturers' brands. But in most urine tests, which work to a NIDA and SAMHSA cutoff of 50 nanograms per mil, you can expect cannabis to still be detectable for between 4 and 5 days after a single exposure usage. Now compare that to up to 30 days after cessation in a heavy regular user, somebody that's been using very concentrated cannabis such as skunk for a long period of time, has built up quite a habit and then stops. The cannabis will be deposited around the body in the fat cells and will gradually be expelled from the body over that longer period of time. So they will still possibly be testing positive up to 2-3 weeks after they've stopped using. The other question we commonly get at this time, it relates to saliva testing and how that compares to urine testing. This is a particular relevance with the new roadside cannabis testing that the police are doing in the UK from March this year. Now, saliva testing for cannabis, particularly the tests used by the police, will only detect cannabis in saliva for between 6 and 8 hours. There are cannabis tests that can detect saliva cannabis residues for 12 up to 18, sometimes 24 hours. The tests we tend to sell for saliva testing on cannabis will detect it for a maximum of 12 hours after use. Now compare that to a urine sample which will detect cannabis for between 3 and 5 days, and in a heavy sustained user anything up to 30 days. You'll see that you can get very different test results depending on the modality of testing that's employed. So we're going to demonstrate this test cassette for you now. So I remove it from the foil. Packed in with the test cassette is a pipette, and to perform the test you simply take the pipette, take 3 drops of your urine sample that you need to have collected separately, and apply those into the sample well. There we go. Now the test will run itself automatically. Osmotic pressure will pull the urine sample along the test membrane that's embedded into this cassette, and if I bring the cassette closer to the camera, hopefully without losing focus, you'll see some pink dye showing in the test window in the central section of this test cassette. Test cassettes vary in design slightly, but basically they've all working on the same format: sample well, a results window, and then at the end you'll have the coding on the test strip shown in a further window. In this case it's THC for tetrahydrocannabinols. And if we just now wait for the test to develop, you're looking for the development of a control line and a test line. All drug tests work to this type of format. The control line has to be present to show that a sufficient sample has been added and the test membrane is run completely. And then you're looking for the presence or absence of a test line at a specified time, and that is what gives you your result. And for drug testing technology, we're looking at a negative binding technology, or a competitive binding technology I should say, which means if there's drug in the sample that's been added to the test at or above the cutoff that's specified by the manufacturer, then that drug will completely bind the colloid membrane at the test line and not allow any of the dye that you saw running along this test window to bind at the test zone. So a positive result is indicated by the absence of a discernible test line at the specified manufacturer's read time. negative test results, you'll get both a test line and a control line forming, and these will usually form quite quickly. Now if we look at this result here, you might just be able to make out that we've got both a test line and a control line showing. Haven't had to wait for the full manufacturer's specified time with the core test, which is 8 to 10 minutes. So we can read this straight away as a clear negative test. Demonstrates that no cannabis at 50 nanograms or above is present in the sample that was added to this test cassette. So there we go, that's cannabis urine testing. Window period for detection is usually after a single one-off exposure between 3 and 5 days. There is some variance there. It's going to depend on quite a few physiological factors such as body mass, urine function, kidney function, and the concentration and total amount of cannabis that's been consumed on the episode that it has been taken. So that's why you'll see demonstrated the window period between 3 and 5 days, and the only way to determine when you go from a positive to a negative test and it's washed out of your system to below the 50 nanograms is to do a series of tests, probably at 2 to 3 hourly intervals over the period that you'd expect it to go from positive to negative.